This conference will now be recorded.
Hello, my name is Ana Conesa. I'm a professor at the University of Florida. And today my talk on this virtual SMRT Leiden conference will be on the tools that developed in my lab to address the functional iso-transcriptomics analysis using long-read data. So you probably have heard about the tools that we have developed in the Conesa lab. Basically, we are working now in three major tools. SQANTI is the tool that we have for quality control of full-length transcriptomes. With IsoAnnot, we tried to give functional labels to isoforms. And tappAS is a tool that we have created to do some statistical analysis and ask questions on the functional relevance of isoform changes. So I will be talking about some of the updates in these different applications. So SQANTI, you know, is a tool for quality control of long-read transcriptomics data. It basically has like two major parts. The quality control section, in which we evaluate the quality of the data, and then we have a SQANTI filter that removes transcripts that are possibly false transcripts from the annotation. So SQANTI creates a large diversity of plots and graphs to assess the quality of your data, but probably what is more relevant of known of SQANTI is the different set of descriptions that we have for the long-read transcripts. These are compared to the reference, and they are full splice match when they are basically the same. The incomplete, you know, have shorter ends in relationship with the reference. Then we have the novel transcripts for known genes. These are the novel in catalog when we have new splice junctions, but using donor and acceptor sites that are already known. And the novel in catalog is where we really have a new splice junction. And then we have all these other categories that are genic, antisense, that are regularly are small transcripts. So SQANTI has followed a number of developments. We first created the tool, in which we have a number of categories. We were creating the report, the machine learning filter that I mentioned before, and we were able to create a curated transcriptome. With the help of Liz from PacBio, she took this development and moved it forward to SQANTI 2, in which she implemented a number of additions, like the minimap mapper, created some additional categories, did the mapping of CAGE and poly-A data, and some additional annotations and quantification functionalities. Now we are working together in merging these two developments into what it will be SQANTI 3, in which we collect all these features, but we also add new ones. Especially, I think that it's important how we are now looking into CAGE and other annotations to improve the quality of the resulting transcriptomes. We are looking very much into how our cold transcripts relate to the reference transcripts. We have more diagnostic plots, we have saturation curves. The new SQANTI 3 will include direct annotation with IsoAnnot lights to create all these functional labels, and we are also working on updates on the machine learning filter. This will be available very soon, but I can already give you some insights of new things that you can do with SQANTI 3. So one of the important features that is now implemented is the CAGE analysis, especially of mostly for full splice match and incomplete splice matches, because CAGE is indicating transcription start sites. And when we do this analysis, we can see some interesting features of both full splice matches and incomplete splice matches. So here you have the distribution of full splice matches that are supported by cage peaks, and then you can see here that most of them have a true cage peak when they are close to the annotated TSS, and then when they are far from the annotated TSS, normally they will not have the cage peak. So that will be for the for one type of the data. So, however, when we look into the incomplete splice matches, the situation is very different, and then in most cases we will not have the fullest the cage peak support, but we can still see a few cases in which even if we have a incomplete splice match, we do have this cage support, so indicating that this is probably real novel shorter transcripts that have evidence of transcription start site. Other important aspects that we identify when they're looking into the diversity of full and incomplete splice matches is how they relate to the reference transcripts. And then we see that many times we have the same reference transcripts that will have the mapping of multiple full splice matches and incomplete splice matches. So these graphs here help you to analyze that, and this is indicating how many reference transcripts will have one or more assigned full of incomplete splice matches. So for example, here we have the cases in which per reference transcript we have only one full splice match of incomplete splice match. So these are this is basically the situation that we want, and these are the large majority. And you can see here in some cases we have incomplete splice matches that will be represented by this part of the bar. So these are incomplete splice matches that are the only transcripts representing a particular gene, but all these genes have only one associated long read transcript. But actually there are many others, so there are all the rest here that can make up to 35–40%, in which we have a reference transcript, but actually with assignments to more than one full splice match of incomplete splice match. For example, here with two, we could have two splice matches, or one splice match and one incomplete splice match, of two incomplete splice matches, and so forth. Okay, so this is giving some indication of redundancy of problems in the collapsing of the data, although actually if we then look at the same analysis but considering only the isoforms for which we have cage support, so a support of the transcription start site, and we have a polydelineation motif that will be indicating the transcription terminated site, we actually see that we also have a number of cases in which the same reference is supported by several long read transcripts. And the numbers are lower if you compare these two numbers, but this is indicative that for these transcripts here, we really have evidence of not yet annotated start of termination sites. Another aspect of SQANTI that is now provided with the report is the saturation curve that is giving the number of transcripts detected as the sequencing depth increases. And then we have these divided by categories, but also by the number of supporting full-length reads. So for example, here when we are considering that the transcript should have at least 2 full-length reads, we are observing that normally the full splice matcher will start to saturate after some point. And this bar here indicates the actual sequencing depth of the sample. This is for a SQL2 dataset in melanoma. So this is the actual sequencing depth, and this is the projection in future. Okay, so the full splice match is kind of having a flattened curve, right? While the other categories are kind of steadily increasing, right? So they are rapidly increasing as the number of reads and the sequencing depth increasing. Okay, so we are very far from saturation here, and this also may be some hints of stochastic, of random, of incorrect transcript code. This is the same plot, but with a different view. Maybe if you look at this one here, in which we have at least 3 full-length reporting reads, you can see that the full splice matches, the number of additional full splice matches as a function of the sequencing depth decreases with the sequencing depth, but actually some other categories, for example, the novel not in catalog, which are the red ones, will actually be increasing. So when you really go deeper in the sequencing depth, you will be recovering more and more of these novel transcripts. This is also because they tend to be lower expressed, but again, could be indication of some kind of stochastic signal. So this is for SQANTI, and as I mentioned, we are working on the release of the SQANTI 3. For IsoAnnot, this is a tool for a domain annotation of isoforms, and what we do is collect a large number of different databases and prediction algorithms for functional domains, both at the RNA and the protein level, and assign the long-read transcripts with all these characteristics that you see here as motifs, repeats, microRNA, RNA binding sites for the RNA, and for the proteins we have PFAM domains, we have signal peptides, all these type of different set of annotations. The result of IsoAnnot is a GFF3-like file in which you have all these different annotations, and that is composed of all these different layers that I mentioned before, in which we have some characteristics for the transcripts, and then we have functional domains and properties for the encoded proteins, and then we also annotate with the gene ontology terms. We have now created this Snakemake implementation of the IsoAnnot pipeline. Is a very, very complex pipeline. We really have to retrieve many different files from reference databases. We have to run different algorithms. All these elements of the pipeline are interconnected, and with the Snakemake, we are able to put all these graphs together, so all this pipeline together, and run this in a kind of controlled way. The good thing of the Snakemake is that if, for example, the process fails at some point and then you restart start it again, it will look into which intermediate fields are present and will resume from that point on. And it can check that everything is finished before it goes into the next step. We also have this now in content environment, so that hopefully people can run this in their systems. So the way it works is that with this structure, or the way we are trying to make it work, is that with this structure, we provide for this species a configuration file that basically indicates the species, and also the URL of the path of the indication of the different sort of data that the pipeline requires to run, so that just changing the configuration file, it could be applied to different organisms. Okay, so now we are completing running this again, so because this is something that we had run in previous version of the different databases. So we are creating this now again, for zebrafish, mouse, Drosophila, and Ariosis, also for human. But we are very happy to have collaborators for other species, since, I mean, this is still a lot of fine-tuning to make sure that the Snakemake pipeline is compatible with other species. So this is the status that we have now with IsoAnnot, and the idea that we have now to combine everything and then to move into the tappAS analysis is somehow represented here. So our idea is that with the long reads or some other type of transcriptomes, okay, so we prefer to use long reads, but other people may use some other sources. and curated data sets, we can use now the IsoAnnot annotation, or even the IsoAnnot Leiden annotation, which is a kind of speeded version of IsoAnnot, to generate an annotated transcriptome that is fed into tappAS. If you don't have the long reads, you have the short reads, you can also use it. At the very end, what you need to do is to create an expression matrix with whatever your reference transcriptome is, and provide an experimental design. Now we are using, for creating expression matrix, we are using short reads data for quantification, but as sequencing depth increases for long reads, we may be able to just use the long reads here. So all these three sets of information are put into tappAS. Okay, if you are using a reference transcriptome, you can just use the built-in reference annotation, so you don't have to go through this process. This is only if you have long reads. But once you have all these three pieces of information into tappAS, different types of functionalities can be run to do this functional analysis. And this is how it looks like. tappAS has a panel in which you can see all the genes and the transcripts. You have here all the buttons to perform the different type of analysis. With this part here, normally you will be able to create graphs of export data, and you can see our transcript visualization functions at this lower panel of the application. Then you have some buttons, for example, here to select, to change columns, to impose filters. Really, the filtering options that we have in tappAS are huge. Okay, and this collects all the information provided here. So which kind of questions you can make with tappAS? Actually, you can make many questions, because it's a very diverse and versatile environment. So you can ask which are the genes that are differentially spliced, and if they are different than the differentially expressed genes. If you have nonsense mediated decay, if you just want to look into coding sequences, so you can also ask if your proteins, or which of your proteins are differentially spliced, in which pathways they are involved. If you now have a particular pathway you want to see, or you may want to see how many of the genes are differentially expressed, differentially spliced, which are both, for example. You can look into the UTRs and polyasides and see how it changes through splicing, or look into differences in binding properties of phosphorylation, transcriptional transmembrane domains, or any other kind of feature that's changed because of alternative splicing, or, for example, whether changes at UTRs are affecting microRNA regulation. This is done through the implementation of 3 modules. So once we have this input data that we mentioned before, we have 3 basic models. The functional diversity will assess the potential of isoform diversity to change functionality of genes. The differential analysis module will do the basic statistics. And then we have a large module with many possibilities to analyze the features. And by features, I mean the functional domains, but also the length of the UTRs, of the length of the coding sequence, and any kind of feature that describes a transcript. The functional diversity analysis basically is evaluating the potential. So here we don't have any expression data incorporated yet, but if you have a gene with 4 transcripts, it will evaluate whether any of the functional domains is conserved or is varying, is changing across transcripts. Okay, so to have an assessment of how diverse the isoforms are in terms of function. So here you have, for example, for one gene, that this gene has a 3' UTR varying region. You can see here. You can also see that the CDS is varying. Okay, so that's in the gray bar represents the coding region, and the lines are the UTRs, and also has variability in the 3' UTRs and polyadenation side, and all these dots here represent functional elements that also will be changing. With this functional diversity analysis, then you can count actually how many genes have differences in annotations, and then get an impression of which are these functions that tend to vary most. For example, here we see that protein kinase domains are more frequently changing across isoform annotations, or RNA recognition motifs are also a frequent category. So this is for the functional diversity analysis. For the part of expression analysis, we have envisioned a way in which we can quantify the expression of the different elements that we want to analyze. And this is actually very simple. And again, if we consider a gene with, imagine these 4 transcripts that are different, okay? So if you look carefully, so this again will be the coding region, and this will be the UTRs. We have here 4 transcripts, but actually we have 2 coding sequences. So this coding sequence and this coding sequence is the same. So the differences between these 2 transcripts are on the UTR, and they are 3' UTR, while these 2 transcripts are again the same coding sequence, but with differences at the UTR, okay? And then we have, for example, these 2 elements that are variant elements, okay? If we quantify the number of reads that map to each of these transcripts, then we can calculate the expression of each element. So the expression of the 4 transcripts will be their own reads, but for example, the expression of coding sequence 1 will be the sum of the reads of transcript 1 and 2, so X1 and X2. The same for 2, coding sequence 2 will be 3 and 4. But for example, for this feature, again, we will have the expression is X1 plus X2, while the feature in red that is shared between transcript 1 and 3, the expression of this feature will be quantified as X1 plus X3. Okay, so in this way we can calculate the expression of any element. Okay, so once we have calculated the expression, then the type of statistical analysis is actually very similar. Okay, so we can evaluate with a linear model whether we have, for that element, we have a change in expression between one condition and another. So here the line represents either a transcript, a coding sequence of a particular functional feature. Okay. When we have these changes, we can also have what we call a switching event. So that means that the major element that is expressed changes from one condition to the other. Okay, so here is the orange, and here is the this is the yellow, here is the orange. And we can also measure what is the magnitude of this change. This is what we call the usage change. So actually, this usage change of isoform, percentage of isoform usage change is a magnitude, is a measurement of the magnitude of the changes between isoforms that is kind of similar of parallel, goes in parallel to what we know about fold change from gene expression. So one of the plots that tappAS returns is a graph like this, in which we represent the fold change of the genes between 2 conditions and the usage change. Okay, so you can see both differential expression and different in alternative splicing. And for example, you can then point to genes, or for example, this one here, that actually does not change in expression between 2 conditions, but has a huge change in usage. Okay. Or some others that's in which the opposite happens. Okay, so then you can understand the difference between the 2 types of transcriptional regulation. So this is an example of how this idea works. Here we have a gene with 4 isoforms. The isoforms, if you look carefully, change in this part and this part. Okay, and then you have with these different alternative included exons. Okay, we have all possible combinations. Okay, here in this part, okay, we have a nuclear localization signal that is created when we have exon exclusion. Then is when we have the nuclear localization signal. Okay. So then we can look at the expression of these different transcripts. And for example, in this case, we observed that in terms of genes, so measuring everything together, okay, so that would be the 4 transcripts together, we actually have no expression changes between the two tissues in this case that we are analyzing. The transcript and the CDSs do change between one condition and the other condition. We have here a strong switch event, okay? This is significant, and we have a strong isotone usage change. This is almost 50% of the expression is already distributed among proteins. But if we then look to this one nuclear localization signal, again, we can see that we have a strong change of inclusion of this functional element with a strong significant p-value, and again, a strong magnitude of change. So this is for one gene, but you can imagine that then we can do this for all genes and all features, and then start looking into which are those features that tend to be more frequently modified from one condition and another. For example, here we are again looking into 2 different conditions in the mouse neural system, and we can highlight which type of domains tend to change in this way as we are measuring, as I mentioned before, okay? And we see that, for example, PFM domains tend to change in a significant way, or microRNA binding sites tend to change as well. Upstream open reading frames are significant, so we can evaluate how strong is the enrichment of these functional changes across, the different experimental conditions. This is again the same on a time course analysis, but basically the same idea. We can evaluate different microRNAs. So this is more on full category based, but this is now for particular functional elements separately. And then we can see that in these cases, we have microRNA binding sites, RNA binding, protein binding sites that change their inclusion in a significant way in some cases, and some other motifs of UTRs between one condition and another, and then start making hypotheses on these functional changes. So as conclusion, well, I have briefly presented here some of the new functionalities in SQANTI 3 to allow the improvements in the curation of transcriptomes. With IsoAnnot, we are able to connect transcript models to functional annotation. tappAS is there to enable a large diversity of analysis tailored to the user's questions, and all this together is what we call the Functional Iso-Transcriptomics analysis, that I think that it will help us to better understand the importance of alternative splicing. And with that, I want to thank you, the students in my lab, of my former lab, and the new people. Most of this work was initiated by these 2 people, Hector, Lorena, and Manuel Tartaguila. Now Angeles is working together with with Fran into the improvement of SQANTI and tappAS and IsoAnnot. Also, Pedro here did a lot of the implementations of the IsoAnnot pipeline. And with that, I would like to thank you also for your attention. And I guess that there will be some time for questions somewhere.